A warm welcome to the launch of the version 25 of the Human Protein Atlas. My name is Matthias Ullen and I am the director of the Human Protein Atlas Consortium. And today we will go through some of the highlights of this very comprehensive new version of the Protein Atlas. The Pan-Disease Blood Atlas, some on the MS-based targeted proteomics, and the very comprehensive expansion of the single-cell resource. The first version of the Human Protein Atlas was launched at the HPA conference in Munich in 2005. 20 years later, we are very happy and proud to launch the 25th version of the Protein Atlas here at the HPA conference in Toronto, Canada. The database is actually one of the most visited biological databases in the world, and on this map you can see the location of all the researchers visiting the Human Protein Atlas, spanning the different parts of the world. The Protein Atlas now contains more than 10 million images, spanning from immunohistochemistry images for tissue profiles to fluorescent images showing the subcellular location, and it is in fact one of the world's largest effort to generate tissue images. So during these 20 years we have actually published exactly 1,000 publications from the consortium, and on this slide we show some of selected publications that was published in the last decade from the group. From version 25 we now have expanded to now contains 9 separate resources, and in the resources we cover all the protein-coded genes. So researchers can explore the tissues in the tissue resource, more specifically the location and expression in the brain. Also a very expanded version of the single cell resource. You can see the location subcellular localization in the subcell resource. We have a very, very expanded version of the blood resource. I come back to this. And then we have the cell line resource, so you can see what is the best cell line for your research. The cancer resource, which is also related to what genes are important for patient survival. We have the structure resource where we used AlphaFold, I come back to this, and a very new interaction resource where we used prediction of interactions using the AlphaFold 3. An important objective of the Protein Atlas program is to annotate all the human proteins. This is done then with 3 main annotations based on expression profiles across tissues, organs, cells and blood. And the first classification is based on specificity, which shows the fold changes between different expression levels. We have a more general tau index that gives you the overall specificity of every protein. And then we also have a sort of an AI-based cluster annotation where specificity and function is based on similarity in expression. So the objective for the future then is to make all of this data AI ready so you can use it in different models to model tissues and cells. In the new version of the Protein Atlas, we have selected a few features that we want to discuss further in this presentation. First, a new comprehensive blood disease atlas using 3 different assay platforms. The use of an AI-based predictions from AlphaFold, both for protein structures and for protein-protein interactions. And then the comprehensive extension of the single cell and spatial transcriptomics resource. The open access resource now has more than 500,000 visitors per month, and we are very proud to have used more than 27,000 antibodies and actually 6 billion sample assays measurements that we show in the atlas. So with that, I'm going to hand over to Maria Bueno Alves that will talk about the new human pan-disease blood atlas.
My name is Maria Bueno, and I'm a PhD student working with the Human Disease Blood Resource. Recently, we released a human pan-disease blood atlas of the circulating proteome, where we profiled over 8,000 individuals with the proximity extension assay, measuring over 5,000 proteins. This dataset allowed us to characterize the plasma protein patterns in healthy individuals across development into adulthood and also associated with aging and disease. By including 59 disease types, we could distinguish proteins that are shared across diseases from those that are disease-specific. All the data that we generated in the study is included in the Human Disease Blood Resource, where users can explore the protein profiles in healthy adults, across development, and also across a wide range of diseases. For each disease type, we also provide summary pages that include the results from differential expression as well as machine learning analysis. In this new release, we have expanded the resource with a new pan-disease dataset that includes over 2,500 plasma samples from over 30 cohorts. And the samples have been profiled using two complementary proteomic platforms, the Olink HT, which covers over 5,400 assays, and the SomaScan 11K, which measures over 10,000 assays. This addition complements the previously available Olink Explore data and broadens the proteomic coverage of the pan-disease blood atlas. Here we can see an example of the newly released data. The protein Col9A1 shows higher level in childhood samples with a decline into adulthood. And we can see that in the pan-disease dataset, but also in the longitudinal data, that we see the proteins from 4 years old until 24 years old. And by having measurements from two independent platforms, we can also assess the cross-platform concordance, and in this case, we see a strong correlation over 0.9. And this highlights the robustness and reproducibility of the data for this assay.
So we're now expanding and introducing a new target proteomics resource to the blood section of the Human Protein Atlas. The goal is simple, and that is to turn the Human Protein Atlas into a resource for absolute plasma concentrations, and to present data that bridge discovery and clinical validation closer together. This will also make it easier for us to explain and calibrate different results across platforms when we know the molar concentration of the protein. We have also updated the plasma concentration backbone using the latest prediction from the Peptide Atlas build and added in-house data to present absolute concentrations of plasma proteins quantified using stable isotope standard protein fragments. And here we're using multiple peptides per protein for extra accuracy and precision. So what will you get in version 25? First, all the plasma proteomics data is based on verified prototypic peptides. Each data point in this release is based on matched heavy standards and allow for absolute concentration determination. So we're not just reporting relative signal intensities for the diseases anymore. We will also present data on a ready-to-run 175-plex cis-calibrant mixture that can be spiked before digestion to enable robust cross-cohort comparability. I would also like to highlight the absolute data from the pan-cancer cohort. That, together with the new data, allow us to explore absolute protein levels across different blood protein classes, such as apolipoproteins, coagulation factors, and proteins from the complement system. All of this is available in this new release.
Thank you, Fredrik. My name is Evelina Sjöstedt, and I will tell you about some of the major updates and changes included in version 25 for the single-cell resource. First of all, the sequence data has been remapped to match the ensemble version 109, which is the one used for the tissue RNA data. And also now 34 tissues is represented. And out of those 34, 4 are completely new and 14 of the old data sets have been changed. So there's been a lot of changes in the data, and this has mainly been used to improve the representation of the different tissue types and also to include some of the missing tissues that we didn't have before. The major update resulted in 1,175 tissue-specific cell types, which is summarized into 154 consensus cell types across all tissues. Some of those cells are only present in one tissue, while others are represented in all of them, such as endothelial cells or fibroblasts. This is the old version to the left, and then to the right you can see the new overview. And although there is an increase from 81 to 154, you might see that the overview is slightly smaller. And this is due to an introduction of a cell type group level. And this is something that we have added now for a better overview. The 154 cell types are grouped into 53 cell type groups. And here you can see them with the 15 color codes used for cell class. This is work done by PhD student Emilio Scarvan. And just to further explain this hierarchy, an example is endothelial and mural cell, here as a class, which includes two different cell type groups, endothelial cells and mural cells, where the endothelial cell as a group include two cell types, vascular and lymphatic endothelial cells, and mural cells include two cell types, pericytes and vascular smooth muscle cells. The main reason for this is to better provide a cell type overview. When we apply the cell type specificity category based on the 53, we get a nice distribution of cell type enriched genes, while for the 154, the majority end up in cell type enhanced. Too many data points leads to a little bit of noise and not as good as an overview. But both these levels are available on the atlas, so you can still explore the level of details. And also on the gene expression level, you have the data points for each the cell type group and the individual cell types. As a final note, I just want to highlight a couple examples related to these major upgrades. To the left, you can see pituitary gland that was previously missing. And now, thanks to the cell type groups, you can identify genes with elevated expression in pituitary endocrine cells. And with the cell type details, you can also explore the different endocrine lineages. Stomach to the right is also a major upgrade. Previous data had limited number of cell types, and this new data includes over 37 tissue-specific cell types, which is then representing 17 general cell types of the stomach. And these are all examples of major updates of the single cell type atlas. And with this, I want to give the word back to Matthias for some concluding remarks.
So before we end, I just wanted to point out that we have AI-based predictions of protein structures, both with AlphaFold 2, as you can see here to the left, where we not only show the predicted structures of all the proteins and all the isoforms, transcript, splice variants, but also you can display where the antibodies used in the protein atlas are binding to the structures. But new in this version is the 3D structures also, or predicted structures of protein-protein interactions using AlphaFold 3. And here you can see the predicted interactions between two proteins, serpin B4 and serpin B3. I am very grateful for all the researchers that have been involved in creating this protein atlas during the last 20 years. I'm also very, very grateful to Knut and Alice Wallenberg Foundation that has funded this program right from the start in 2003 into the present days. To end, I want to show you an image of Stockholm where you can see the Science for Life laboratory to the left on this image at the Karolinska Institute campus. And with that, I want to thank you all for listening to this presentation. Thank you very much.